Wi-Fi.
Very good morning to one and all present here. Respected Director, ICR Central Tobacco Research Institute, Dr. D. Damodar Reddy sir, and today's honorable guest speaker, Dr. D. L.N. Rao sir, ICR Emeritus Scientist, retired, ex-project coordinator, AINP SBB, IISS Bhopal, and head of the divisions, research stations, scientific, technical, and administrative staff from CTRI and other faculty from all the institutes of the nation. I request Dr. Suman Kalyani Madam to give floral welcome to dignitaries. As a part of ongoing celebrations of Azadi Ka Amrit Mahotsav, commemorating the 75 years of India's independence, ICR Central Tobacco Research Institute, Rajahmundry, in collaboration with Indian Society of Tobacco Science, Rajahmundry, is organizing lecture series. This is the 5th lecture series the institute is organizing. First 4 we have conducted online, and this is the first offline lecture. Requested guest speaker Dr. Rao sir kindly accepted to deliver a special lecture on very important topic, soil ecological stewardship, soil health and microbial inoculants. This session is chaired by Director Dr. D. Damodar Reddy sir. We are very grateful for your glorious presence in CTRI sir. Okay. Sir, we are very grateful for your glorious presence in the institute, sir. Thank you, sir, for accepting our invitation and for delivering today's very important lecture. Now I request our director, sir, to give introduction of the speaker.
Honorable Dr. D.L.N. Rao garu, who is with us as chief guest for delivering a lecture as part of the Azadi Ka Amrit Mahotsav celebrations of this institute. My colleagues, senior colleagues, heads of divisions, and also the scientists, technical staff, and other members. We also have presence of other colleagues from research stations who are participating online. Very good morning, and it's always a pleasure to stand before you and introduce a person with whom I worked for so many years, maybe 7-8 years, sir. 7-8 years there in Bhopal, Indian Institute of Science. And truly speaking, Dr. D.L.N. Rao garu needs no introduction actually. He is a very popular scientist across the country, not only in India but also abroad. And it may not be an exaggeration to say that he is genius par excellence as far as the soil microbiology, biofertilizer, soil biota, soil microbial diversity, soil enzymes, all these important areas. Please keep yourself mute for some time. I say he is genius par excellence because he belongs to a generation which has really concentrated on the basic research, in-depth understanding of the soil biota and their applications as biofertilizers. But of late, probably we are not paying much attention that the field of microbiology, soil microbiology in particular, deserves to be receiving so much of attention, but we are not paying it. Maybe because of the shortage of the people who are interested in hard work. Actually, microbiology needs a lot of hard work. It's not that superficially we can get along. But microbiology is such a subject, it needs a lot of energy, time, and efforts. So he belongs to that kind of generation. And even today, wherever he goes, people recognize him by his hard work, by his achievements in the research field. And more than anything else, he has contributed a lot to the for the benefit of the youngsters, sir. Here we have got a lot many youngsters who are specialized in different disciplines. Probably they must have not come across your systematic work that you have done in the field of nitrogen fixation and biofertilizers in particular, and also soil ecology as a whole. So, and fortunately, the fact is that he belongs to this area only. And though he has been working over the years in most parts of the Andhra, Telangana, the North India, but he has now joined us here in this a place which is very familiar to him. Dr. Rao actually graduated from Delhi University. Subsequently, he completed his post-graduation, MSc and PhD from Indian Agriculture Research Institute, New Delhi. Then his main focused research area is whole soil microbiology. You can say whole soil microbiology, particularly nitrogen fixation in legumes, be it annual crop or a perennial legume. And then he did a lot of work on soil organic matter transformation, nitrogen transformation, and then soil enzymes, their role in the soil fertility maintenance, and also soil health management. He did postdoctoral research at the University of Kent, UK, and he has been visiting many countries. And he was a visiting professor, research fellow at University of London, as well as the University of Sussex in UK. He has been, we have got a tendency to shift our research areas. But over the years, maybe more than 3.5 decades, and until recently he retired as a professor of eminence, he has been focusing on the soil microbiology, their role, sustainable agriculture, all these things. Then if you look at his background, research background, he joined at Central Soil Salinity Research Institute at Karnal in Haryana. And he joined Indian Institute of Soil Science in 1998 as principal scientist and also the project coordinator of the then AICRP on biological nitrogen fixation, and subsequently which was changed to All India Network Project on soil biodiversity and biofertilizers. Yeah, there he worked for about 2 decades. Then his major contributions lie in assessing the genetic diversity of the rhizobium, then soil enzymes, soil genomics, microbial diversities, and then more than understanding these things and transforming that knowledge into practical solutions, he has contributed a lot to the creating awareness about the biofertilizer technologies and also the landmarks which we landmark achievements. Biofertilizer uses in different thermal regions, like in high temperature regions, low temperature regions, temperate tropical conditions. These kind of contributions generally we don't come across. People work in only one specified ecosystems. There they are specialized, but he has got vast knowledge, wide experience of traveling to different countries, and he has done a commendable job. That is not that I know him, that's why I'm not boasting, but it is recognized and recognized by one and all across the country, not only in the country but also abroad. He has published more than 150 research articles. That itself is a big achievement. And he has participated in many collaborative research projects, international projects, and also he loves to lecture. And you will realize why he loves to lecture and why people love him, asking him every time to deliver a lecture. Whenever he goes there, definitely, you see, this time around, he was supposed to come to some other assignment, but immediately it struck my mind, why can't we take advantage of this, his visit to this place? And immediately I asked, he was kind enough to agree within no time, within minutes, on telephone only. He said, yes, why not? We will definitely deliver a lecture, and that's how it has got materialized. The most significant achievement towards end of his career is that he has ensured that as an emeritus scientist of ICAR, established as well as headed the Indo-UK Nitrogen Fixation Center. That is now his end service achievement, you can say. It's a creation, it's a big achievement. This has been done under the DBT Newton Baba Fund. By utilizing the DBT fund, he has created one India-UK Nitrogen Fixation Center. Dr. Rao is well known. As I said, he is a fellow of the National Academy of Agricultural Sciences, New Delhi, and also the fellow of Indian Society of Soil Science, New Delhi. He has won dozens of awards, and to name, I think we will have to. There is no point in listing them now. Several awards. He has played a very crucial role in preparing the. See, we also prepare the perspective plans for our institute, but he involves himself in preparing the perspective plans for different agencies, for different institutes also. And he has been working, providing the guidance on by being on the RACs and also QRT teams of many ICAR institutes. Busy person rather. He is busy in science. Many people are busy in many other activities, but he is busy in science all the time. Towards the end of his, almost now he is 70 plus, sir. 68 also, he is very active. See, while walking also, he will be walking more faster than us. So you can imagine how active he is. 67 plus. His special interest, other than research anyway, he is well known person in research. He loves to take the lectures, and he worked as the adjunct professor, and he was a visiting professor for many other countries. He teaches even now. He teaches as an adjunct professor and takes keen interest in mentoring as well as nurturing the young people, young talent, whoever are interested in research. By listening to his lecture now, just we are going to listen. After that, many of may also get motivated, and probably your lecture is going to inspire themselves. Certainly, I am 100% confident. After listening to his lecture, many of you get inspired to do some research, the kind of research, if not what kind of research he has done, at least part of that research, if you start doing, that is going to be a good achievement for the institute and also for your personal career. So without wasting much time, without unnecessarily talking so much, because you are eagerly waiting, and this is going to be a different lecture. I am 100% confident. You have not listened to his lecture. It is going to be a different one. You may be expecting something of this. You will be failed in your expectations. You will be going to see a different kind of lecture. At least I know him personally. 6-7 years I was there in Bhopal along with him. You will be always looking, research has to be perceived in this context. So, sir, without wasting much time, I thank you very much, sir, for having agreed to come over here. Physically, that too during the Corona pandemic time, he has agreed readily to be with us personally and deliver this piece of lecture. Thank you very much, sir. Thank you. You can deliver from here also. Thank you. Meanwhile, I'll just he'll be coming within 2 minutes. So he was there in Bhopal, Indian Institute of Soil Science. I was also there. Of course, he used to be in different division, but at least I used to personally go to him, interact with him. Some of the words that you will listen from me sometimes, you will also listen from him. I have learned a lot many things from him because he is a hardcore researcher. During the office hours, you will never sit in one place. You will be going to laboratory, you will be going to some other division. Got it, sir? Yeah. So this is we are fortunate enough to have him here, and today we will be going to be enlightened by the soil rhizosphere. He is going to talk about ecology, everything, sir. Sir, thank you very much. Yeah.
Thank you very much, Dr. Damodar Reddy, or I said, dear Dr. Damodar Reddy. I've never had such an effusive introduction as a speaker, and it's very embarrassing. And I hope I can live up to the expectations which all of you have been. It has aroused a lot of expectation amongst you, and I am sure that I hope that I will be able to come up to your expectations. Can you? Yeah. 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 Thanks very much. Now, the reason why I chose this particular topic, or let's say the title for this topic, is something which I will let you know right at the last slide of mine. This is the word is soil ecological stewardship. And the word steward has been used because steward is one who takes care of you, whatever you want, when you are in an aeroplane or anywhere else. So that means what we need to do is to actually take care of soil the way a steward takes care of us. And when I said soil ecological stewardship, that means to take care of soil ecology. So it's something like what you call a soil care. This is the word which is used in many places, you must have heard. And we are now celebrating the International Decade of Soils. The International Union of Soil Science has declared 2015 to 2024 as the International Decade of Soils. So that shows you what is the importance which the United Nations has given to soil. This United Nations International Decade of Soils. And last year, on the World Environment Day, 2021, yeah, I just had kept it slightly away. Am I audible? I hope I am. It's a very small room. I just kept it away so that it doesn't interfere with my vision. So last year, the World Environment Day, they had chosen the theme of ecosystem restoration. So that also again tells you that something is seriously wrong with our planet, and we need to do something very urgently. So these are the two things which tell you how important soils are. That the United Nations has declared a whole decade as International Decade of Soils, and an International World Environment Day as International Ecosystem Restoration. Now, what I'm going to talk about is centered around the state of the environment. I'll tell you what is the state of the environment, what are the United Nations Sustainable Development Goals, and then talk about soil health, and then ecological solutions to that. Now, this picture, which all of you must have seen so many times, this is the population of India, which is at the top. We have already overtaken China at this point of time in 2022. And then, not only we are the most populous, you can see how much more population we are going to be. Look at the yellow line. Whereas China's population is declining, ours is going to stabilize probably at 1.6 billion. So that means it shows the challenges which our country has to face, which other countries don't have to face. The others, like the European Union and the America, which you see in the USA, they are far lesser than us. So this tells us what is in store for us to come. Just this one picture is quite a scary picture actually to see. Another picture which is very scary and is what are called as planetary boundaries. There are certain planetary boundaries which our human activities, we are not supposed to cross those boundaries. This is what is called as a safe zone. We're not supposed to cross the safe zone. Now, if you see this circle, this globe that I have shown, and if you look at the green cycle here, this is the safe zone. We're not supposed to have actually crossed this. Now, just see it in a pictorial manner. Just as I showed you that population, how much we are grown, and that's quite scary. Look at this rate of biodiversity loss at the top one. It has gone out of the circle far beyond that. And this is in 2009. That means 12 years ago, the extinction of the population was so fast, you're losing species even in Not in terms of days, but in terms of minutes, 3 species per minute or something like that. I don't know the exact figure. So it's already gone. So you can't control that rate of biodiversity loss. It's just gone. And climate change. And the second one is nitrogen cycle, interference with nitrogen cycle. This is the one. Again, you see the nitrogen cycle has gone out within terms of oxides of nitrogen production, nitric oxide production, ammonia losses and others because of use of high fertilizers. Again, it has gone. That means we get, so that means straight away there is a message there that we have to reduce the amount of nitrogen fertilizer. And the second thing is, third thing is climate change, the third one. And here is climate change. So already we have crossed, but then we have to take urgent measures, and that's why you know the various measures to curb climate, which India is signatory to various kinds of treaties to reduce our emissions of methane, carbon dioxide, oxides of nitrogen, and all these greenhouse gases. Now, here you see a smaller one just by the side of nitrogen. Here you see phosphorus. So still within the green cycle, still within the green zone, but we're just about to cross interference with phosphorus cycle and ocean acidification. So this is ocean acidification that you see here. Ocean acidification is also here. Now the ocean acidification happens. Imagine the change in pH of the oceans. That's what is happening. That means the kind of, when the rivers goes and meets the oceans, so much are the pollutants in the rivers that they're actually changing the pH of a vast body of water like oceans. They are becoming more acidic, and that's because of fertilizer usage. There are 3 others which are in danger, which is change of land use, that means from forestry to agriculture, global freshwater use, and stratospheric ozone depletion. Now this is something which is which are also in danger, and not quantified are atmospheric aerosol loading and chemical pollution. So these are a picture by 2009. I don't have an updated picture after this. These are the planetary boundaries that must not be transgressed and could help prevent human activities from causing unacceptable environmental change. On top of this, there are new concerns about global pandemics. There are global health emergencies, COVID-19, which is as grave as climate change. And there is another one which is antimicrobial resistance. You must have all heard about antibiotic resistance. This is a silent pandemic, more scary than even COVID-19, because deaths are happening every day as a result of antimicrobial. And this is as grave or even more grave than COVID-19. Only thing is it's a hidden danger. So therefore, 2 years ago, the United Nations declared November 18 to 24 as the World Antimicrobial Awareness Week. That consists of preventing misuse of antibiotics, because antibiotics are prescribed indiscriminately by doctors, saving antibiotics, producing food without antibiotics. In the animal industry, a lot of antibiotics are used. Ethnomedicines, that means go for medicines which are of an ethnobotanical origin. And then composts, which are suppressive composts. Instead of using fungicides, and then you use composts which are suppressive, which will suppress plant disease. So there have been national action plans to have One Health action plans, and here is the role of soil scientists and microbiologists is paramount. This is a slide which I want to show you about soil degradation. Globally, if you look at the map of the world, and these are the red zones, which are the soils that are very degraded, and you look at the soils, the map which is yellow, which is degraded. The white areas are the stable areas, and these are gray areas are without vegetation that you're getting. Even a child can see from here, from this picture, that the soils are degraded everywhere. There's hardly any area in the world today where you can say that the soils are not degraded. Completely, they are either, it's a question of either more or less. More than 40% of the world's area is degraded. So natural land resources are being degraded through soil erosion, salinization of irrigated areas, dry land degradation from overgrazing, over extraction of groundwater. This is a big problem in areas like Punjab, Haryana, even Tamil Nadu. Growing susceptibility to disease and build-up of pest resistance favored by the spread of monocultures and the use of pesticides, and loss of biodiversity and erosion of the genetic resource base when modern varieties displace traditional ones. Before I go on to my main lecture, and we are talking about the background of the state of the environment, water is obviously the most important, and water pollution is a major problem in the global context. Globally, at least 2 billion people use a drinking water resource that is contaminated with feces. And contaminated water can transmit diseases such as diarrhea, cholera, dysentery, typhoid, and polio. This is well known to all of you. Contaminated water drinking is estimated to cause about 500,000 diarrheal deaths each year. 500,000. And a staggering 4.2 billion people across the world still live without access to toilets. Almost out of the world's 7 billion population. Much of that was in India. Until now, the situation has now been remedied through our action plans. So therefore, in June 2021, when I mentioned that the United Nations then declared as a World Environment Day, they made a statement that there has never been a more urgent need to revive damaged ecosystems than now, according to the United Nations Environment Program, which launched the Decade of Ecosystem Restoration. So I mentioned about the Decade of Soil Health from 2015 to 2021. So now from 2021 to 2031 is the Decade of Ecosystem Restoration. And the world message by United Nations Secretary General Antonio Guterres, I think one line is very, I think, telling. Science tells us that these next 10 years are our final chance. That's a very scary statement. It's our final chance to avert a climate catastrophe, turn back the deadly tide of pollution, and end species loss. And I don't know about Rajahmundry, but I can tell you about Bhopal. At least, and all of you would agree, that the climate statistics of this year has shown that March has been the hottest March perhaps ever recorded in India, in Delhi, and we could feel it in Nepal also. And this was, in 100 years of ever record keeping in India or even more, this was the hottest March that we have seen. Therefore, the United Nations on 1st of January 2016 agreed on 17 Sustainable Development Goals. This is the 2030 Agenda for Sustainable Development. These goals replace the Millennium Development Goals. As you know, they were MDGs, they replaced them. And these were adopted by leaders in September 2015 at a historic UN summit. It provides a shared, and what are these goals? I would not go into the details. You know all of these. You would have seen this picture a number of times. This is poverty, ending poverty, zero hunger, good health and well-being, quality education, gender equality, so on and so forth. You know all these important goals. There are 17 goals, 169 targets, and 6 national indicators. This provides a shared blueprint for peace and prosperity for the people and the planet. At its heart are the 17 Sustainable Development Goals, which are an urgent call for action by all countries, developed and developing, to do these things in global partnership. India is monitoring out of these 17 goals. 13 in 2019, we monitored 13 goals. In 2021, which is the latest report, we are monitoring 17 goals, 70 targets, and 159 indicators. The rates are given in 3 categories. Below 50, you're a failure, you're still an aspirant. Between 50 to 64, you're a performer, and you're a front runner only if you're in 55 to 69. The overall score of India is now 66. So in 2021. Yeah. India ranks at 117 out of 166 countries in 2020. And in 2019, it was at 115 out of 162. So we need to do more and more. And that is why I think the government's efforts are more towards sanitation, towards drinking water, quality education, gender equality. We are lagging behind in a couple of them, like gender equality is one of them. I have not gone into much detail. I have given lectures only on these goals separately. So I have just taken the final conclusion from the United Nations. So far as India is concerned, what do we need to do more? Where do we need to do more? Foster innovation. This was the key message that in India, now this is a message for young scientists, that where are we lagging behind in terms in the Sustainable Development Goals, and what do we need to do in the future? Foster innovation. Innovation, we are poor in innovation. Number 2, clean alternate green energy. That means we need to reduce the consumption of petroleum gas. We need to go more towards alcohol-based industries, or maybe hydrogen-based industries, green hydrogen, whatever. We need clean alternate green energy. We need novel waste processing technologies because we generate a lot of waste. We need novel waste. We need clean ecological solutions. So that is why this is where I'm coming from. Basically, my lecture is coming from here. We need clean ecological solutions. So therefore, we need to encourage bioproducts to reduce fertilizers. I've already shown you nitrogen, how much far it has gone. Phosphorus is just about to go. And we need to reduce pesticide consumption, and that's where all of you come in. And then we need to reduce groundwater withdrawals. I've already mentioned that. It's unsustainable groundwater withdrawals. So we need to conserve more water. And then we need afforestation technologies, climate change. So what I wanted to show by this slide is that we in the ICAR are very much there. This is what we are very much there in providing solutions to the problems which are noted in the United Sustainable Development Goals in order to achieve and then increase our rank, which is, we are not so much interested in achieving our rank, that's all right. But the important thing is to save our planet. And these are, there's nothing new here for all of you. All of you are aware of this, but this brings you into a focus. This one slide, you can take a picture if you wish, that this is where we want to concentrate. And this is a slide which will apply to all ICR institutions, whether it is CTRA, whether it is CSSRA or IASS. This is, and we have got a role to play in each one of them. So I end my first part of my lecture here. Before I go into the specific part, this is about the general background. Before I go to the second part of my lecture, which is on soil degradation and soil health. So soil degradation is a major global concern. I've already said that. Soil erosion. Now, when soil erodes, which is the most important, let's say major contributor of soil degradation. And in India, if you see, 324 million hectares is our land area, and our reporting area is about 300 million hectares. Out of 300 million hectares, something like about 120 million hectares are degraded in one way or the other, whether it is through soil erosion, whether it is through salinization, acidification. And when soil erodes, then everything is gone. There's no question of soil degradation. When soil erosion takes place, then it's gone. There's nothing. The soil itself has gone. So soil degradation is a major global concern. Soil erosion, diminished soil biodiversity, even within cultivated soils, due to various kinds of anthropogenic interventions, low soil organic matter, and nutrient depletion are among the leading contributors to impaired soil health and crop yield. So these are the four major reasons why we need to focus on. And poor soil health in turn leads to low input use efficiency. When we say that nitrogen use efficiency is very low, in the case of rice, it can be as low as 30-35%. In the case of wheat, it can be 56%, but certainly not more than that in any other crop. One of the reasons of low input use efficiency is because of low soil organic matter content. And that's itself what it is because of poor soil health. When there's poor soil health, the soil cannot build soil organic matter. And when soil health is poor, it leads to decreased ability to withstand environmental stresses. So if there is a drought, there's no rainfall. If the soils were green manure in the previous season, or if there is more organic matter content, the soils have a greater ability to hold water. There's more microbial activity, more soil health. So the soils will be able to withstand stress better than a soil which has a lower soil organic matter. And decreasing factor productivity. So when you say about decreasing factor productivity, it's also responsible. Low soil organic matter content is also responsible. Low soil nutrient also responsible for those. Large majority of India's cultivated soils face multiple abiotic stresses. So these stresses are not which are coming singly, whether it's pH or salinity or low organic matter content or low nutrients. They're not coming singly. They're all acting together. So then you have a synergistic effect, negative synergistic effect, which adversely affects soil health. And global warming will further exacerbate this problem. So already the soils are degraded, already the soil health is poor, and global warming is only going to worsen the problem. This is the latest definition of soil health. There have been various definitions of soil health. So I'm not going to go into the old definitions. The Intergovernmental Technical Panel on Soils, there's an ITPS, in September 2020 gave a very simple definition of soils as the ability of the soil to sustain the productivity, diversity, and environmental services of terrestrial ecosystems. Why I have put this in red words? Because the older definitions of soil health, and Damodar will remember all those definitions, they never had this word of diversity in them. This is the first time soil health has been defined in a new way, stressing or bringing the importance of ecological diversity into that. Sustain the productivity, sustain the diversity, and environmental services. What are the environmental services? Whether it is cleaning up of the soils, cleaning up of the waters. The waters are essentially purified when they go through the soil. The groundwater becomes purified. Filtering, buffering, and transformation action of the soil is what cleans the soils. We all know that. So a healthy soil is biologically active, containing a high level of biodiversity, and in which the physical, chemical, and biological processes are functioning optimally. Soils and their biodiversity thus play a critical role in maintaining soil, plant, animal, and human health. So it is a continuum. We should never talk about soil health alone. The plant health depends on soil health, animal health depends on plant health, human health depends upon the health of all these. It's a continuum and not a single. This is a slide which I have added today morning because of a certain reasons which you will come towards the end. Because when we're talking about sustainability or soil sustainability, so not only do we need science, the science needs to go to the farmers, and then it needs to go towards the policy development of the country, whether it's on fertilizer usage or anywhere else. So we need to reduce the fertilizer hotspots wherever they're occurring. It's not as if India has got an average fertilizer consumption of maybe, I think it's around 110 or something like that, approximately kg per hectare. It is not as if it is 100 kg across the whole country. There may be some areas where it's very low. So it can be from 0 to 190. So those are the fertilizer hotspots. And now the fertilizer hotspots globally, which used to be earlier in the West, have now shifted to India and China. India and China are now the major consumers of nitrogen fertilizer. The West has realized that and reduced their nitrogen fertilizer. So the situation has just reversed now. So for this, we need to reduce the inorganic nitrogen fertilizer usage, improve nutrient use efficiency in order to reduce emissions. And for that, we need to go for strategies like integrated nutrient management. Before I go to the solutions here on the left-hand side, this was a slide which I used for the Prime Minister Summit which I will come in the paper summit. So therefore, I thought I'll put this important slide. I'll show you in my conclusion also. This is a slide from Bennett in 2013. I want to show you a very good slide. This is from 8000 BC when agricultural revolution started and agriculture started to 1950, which is the beginning of Green Revolution in the West, to 2010, which is the beginning of Evergreen Revolution, when they have realized the ecological consequences or destruction of that and they have changed their method. And this is going towards 2050. Now, if you look at nitrogen, now the consumption of nitrogen, phosphorus, and the increase in cereal production has happened with a parallel increase in nitrogen and phosphorus consumption. And this has led to a global population like this. While this has happened, while parallelly there was increase in nitrogen and consumption and increase in population, the amount of agricultural nitrogen fixation has actually fallen after 1950. Now, if you remember, all of you must have read at some point of time that, at least I read when I was studying in PUSA and ICAR at that time, we used to say that biological nitrogen fixation globally contributes about 140 million tons of nitrogen per hectare. And at that time, chemical fertilizers manufacture was only 50 million tons. So we used to say that biological nitrogen fixation is 3 times more than chemical fertilizer. But now the situation is reversed. If you look at the biological nitrogen fixation, it has come down to, as I said, it has come down, as you see, it is something like about 100 million hectares. So 140, it has come down to 100. But chemical fertilizer consumption has gone up from 50 to now it's about 125 or 120 or even slightly more. So it's just the reverse. So we are consuming, as a result of agricultural nitrogen fixation is reduced. Mycorrhizal response also has come down. Why? Because we're using more and more phosphorus fertilizers. And loss to pathogens is something which goes up and down. So this is also a very dramatic picture which shows you that we need solutions while the population is increasing, we need to use chemical fertilizers. We cannot do away without that. We have seen the consequences of doing away with chemical fertilizers in countries like Sri Lanka and others where you have seen problems with doing away completely with chemical fertilizers. So we need to somehow use them very intelligently through integrated nutrient management or improve fertilizer use efficiency. And what are the solutions? Is only through improvement of biological nitrogen fixation. Either it has to come through more like annual legumes, through rotations or through intercropping, or perennial legumes in traditional agroforestry systems where you've got nitrogen fixing trees. Use microbial inoculants like Rhizobium to fix nitrogen, or plant growth promoting rhizobacteria like Actinobacterium or Actinomycetum. And then monitor soil biological health, for which you need to study soil microbial ecology, biochemistry of soil, organic matter, soil environment. So these are the solutions which are here. In fact, this summarizes my whole lecture. I thought, let me summarize my lecture in the beginning itself on what are the solutions here. This is a slide which is from Bhanpur. There's a dump near Bhopal. Bhanpur, you know Bhanpur very well. When you travel from my institute to Ayodhya Nagar, on the way you will pass the municipal dump. And I'm sure you have these municipal dumps all over. There's just a slide to tell you that what happens actually when you dump these things. This is one of my colleagues' slides from Division of Soil Biology, Dr. Mandal. And then when we took soil samples from adjacent farm and then also on the dump where the plants are growing and studied the bacterial numbers, look at respiration, which is carbon dioxide evolution, dehydrogenase activity is another indicator of soil health, and fluorescein diacetyl hydrolysis. See the drastic reduction. It's not some 5% or 10% or 15%. Just look at the drastic reduction in all these indicators of soil. So this is something that, and soil is a sink for everything. Ultimately, it is soil which has to purify everything, whatever compounds we're using. And in our life, probably we used to say we're using about 50,000 organic and inorganic chemical compounds that we use in our daily life, whether it's cosmetics, detergents, sprays. There's so many paints, and maybe even more now. This figure of 50,000 I'm giving is more than 10 years old. There can be even more. So everything, ultimately, soil is the ultimate purifier. So if you want good soil health, so good soil health is the key to productive soils, clean water, we know. This much we have appreciated. But what we have not appreciated is that good air quality. This is something which we never think of. You know, we talk about soil quality, we talk about water quality, we have never talked about air quality. We think that only if you are besides staying by a paper mill, maybe you can smell the emissions which are coming out. But even air quality, whether in terms of methane, whether in terms of other organic volatiles which come up, there are so many of them which we are not even aware of. They may be in ppb quantities. So all that is ultimately dependent upon good soil health. So when I said that microorganisms are responsible for sustainability, sometimes because we cannot see microorganisms, and we thought microorganisms can be seen only under a microscope, we think, oh, they are important, we know that. But unless you have a number in your hand, you cannot actually realize how important a particular species is or a person is. It's just like a GDP, unless this GDP is something which you can catch and say. Now, what is not known is that the number of cells on this planet has been estimated at 5 into 10 to the power 31 prokaryotic cells, bacteria. But what they make up in size, because they're very small, but if you add up the weight of these, they weigh something like about 50 quadrillion metric tons, which represents about 60% of the total biomass of the planet. That means whether you're talking about forests, whether you're talking about plants, whether you're talking about insects, whether you're talking about whales, whether you're talking about snakes, all the entire plant animal kingdom, all plants and microbes put together in the soil, in the water. If you just weigh them, the total of that total biomass is microorganisms. This is something which is never taught in textbooks. It's not there anywhere in books, but this is the actual picture. That means they weigh far more than all of us put together. And we all read in our textbooks in school that plants produce oxygen, right? And then during night, and then they during daytime, and evolve carbon dioxide at night. But bacteria includes cyanobacteria, blue green algae, which are in the oceans, right? They produce about half the oxygen on the planet. So when we say plants produce oxygen, we should say plants and bacteria both produce oxygen. So this is where we need to correct our textbooks for our young children. And what are the most important functions of microorganisms? There are several. But then if you just put 4 of them, I've just put only 4 of them in terms of which are most important. Most important is obviously the decomposition of organic matter. So when we everything that's going into the soil is decomposed in microorganisms, which leads to soil organic matter formation. I have written soil organic matter formation with a particular reason why I wrote that. I'll come to the end of towards the end of my lecture. Because it is decomposition of organic matter which forms soil organic matter. Nutrient cycling. So all the nutrients that are released in the soil organic matter is released, including gaseous emissions, whether it's methane or oxides of nitrogen. And then soil structure. The polysaccharides which are produced in microorganisms, the build of the soil aggregates, biofilms, and pore networks. These are again formed by microbial activity. And then regulation of pests and diseases. I already mentioned all the antibiotics and everything. So that's it. And plant health, if soil health is good, plant health is also good. So plant health is tied up with soil health, the microbial communities in the rhizosphere, and the plant microbiome. So now it's realized that the plant microbiome is responsible for plant health, but where does the plant microbiome come from? Does it come from the seed? Yes, some of it does. But where does the majority of the plant microbiome come up? It comes from the soil. As the plants grow, they acquire the microbes through the roots and they go into the health. So if you want to have healthy plants, the soils also need to be healthy. So now while the DNA is called as the primary genome, so there's a secondary genome of the plant, which is now called as the second genome, and Parker would know that it's called second genome because it's coming from the microbes which are present in the plants, coming from the soils, the second genome. Various mechanisms influence plant productivity, whether it's nitrogen fixation, mineral nutrient solubilization, nutrient acquisition, production of hormones, vitamins, enzymes, antimicrobials, induced systemic resistance. This slide was very detailed. It had various things which I cut out just to put only the main mechanisms by which microbes influence plant health. So now to conclude this second part of my lecture, food production, mitigating climate change, and ecosystem services are thus closely intertwined and all rely on soil biodiversity. Therefore, we urgently need to take measures for ecological stewardship of terrestrial ecosystems that builds on improvement of soil biodiversity in order to reverse soil degradation, sustain agriculture, forestry, animal production, fisheries, and etc. So now we have now appreciated through this whole, in the first 2 parts of our lecture, how soil biodiversity is important, how the climate is changing, how the environment is degrading, and how biodiversity has got a very important role to reverse soil degradation. We are now so far talking about maintaining. Now we have to use it to actually reverse because some of the systems have just gone way out. As a group, soil microorganisms are mostly unknown. As you know, when you culture microorganisms on a plate, you hardly recover 1% of the diversity. 99% of the microorganisms, you do not know what they are because it's not possible to culture the microorganisms because we do not know what are the nutrient needs. I mean, it would be very simplistic to assume that you thought, you take an agar plate and put some medium and then you would get all the microbes. No, nature was not devised like that. There are so many secrets in nature. These cannot be replicated in the laboratory so fast. So the estimated number of species, if you see in bacteria, hardly we know 1%. We hardly know 1% of the bacteria. We don't know 99% of them. Fungi, only 1 to 2%. Protozoa, 7.5. Nematodes, ants, termites, columbola, mites, so many others. And then if you take a soil, this is just a pictorial diagram from one of the textbooks. Soil air is about 25%, water is about 25%, mineral particles about 45%, and if you take organic matter 5%, which is very, very, this is from a Western textbook of a forest soil. Otherwise, in India, you can assume this is about 0.5%, so you can just divide that by 10, 0.5 to 1%. What is important is, while the living organisms are about 10% of the soil organic matter, and roots are about 10%, the biomolecules which are produced by microbial action, which I'm going to talk about, and I was going to talk about the characterization of biomolecules that are also the same. But it is these organic microbiomolecules which will be characterized. I'll be showing one slide on that. As you know, these size, either you can see them, large microorganisms like earthworms and roots also are called as microorganisms. Meso, size like springtails, columbola and others, and microbes like yeast and bacteria, which can be seen on the microscope. Now this is the same thing, which shows that the number of microorganisms in 1 teaspoon, if you take, in bacteria can be up to a billion. There can be up to a billion microorganisms, and this is the biomass. So these are all slides which all of you must have seen in one or the other presentation. So I will not show in this here, but we'll just go further. This slide I'm going to show you what are the 3 most important reasons which control biodiversity. Because ultimately we are interested in practical information which you can pass on to the farmers, which you can implement. One is climate, other is soil conditions, other is resource quality. These are the 3 controlling factors of. Obviously, more the moisture you have, more microbial action. In the dry lands it will be less, and so a moderate temperature and a moderate moisture content where microbial activity is best. Climate influences microbial diversity. Soil conditions, pH and habitat structure. Habitat structure, I'm talking about pore structure, a neutral pH. And resource quality. What is the kind of vegetation? Is it a legume? Is it a grass? Is it a forest? And then what is the management practice? Yeah, are you doing zero tillage? Are you having intensive tillage? Are you applying too much fertilizers, pesticides? So these are all those which influence management practices. Second part I said about microbial inoculants. So microbial inoculants, this is slides from my Indian Institute of Soil Science, all our work. Various kinds of mechanisms like nitrogen fixation, whether it's symbiotic nitrogen fixation by rhizobium or non-symbiotic nitrogen fixation by Arthrobacter, phosphorus solubilization or mobilization from various phosphate compounds, potassium and micronutrient mineralization through acid production. Hydrophores are a class of compounds which are produced by microbes which sequester iron and other things. And these are important in competitive inhibition to reduce the iron, make it unavailable to the competitors if it's a fungus. Antibiotics production is a direct mechanism. Hormones production, whether it's indole acetic acid and gibberellin production, inducing immunity by systematic alleviating stresses. So these are the various mechanisms. These are the various groups of microorganisms, whether it's mycorrhiza, whether it's Saccharomyces, which are used. We screen them, and then these are the control plants. These 2 plants on the left are control, and these are inoculated. So this is how you select a microorganism. Inoculate it in small pots, and you see when you inoculate all these ones, they have got more root biomass and more shoot biomass. So these are, and then you so on and so forth. You can take them to the next stage. So, and then you take them to the field. When you inoculate them, this is a check strain. These are the improved. This is pigeon pea. So when you inoculate them, you have this improved strain. Inoculate them so that they have more nodules. For example, this is, as I said, pigeon pea. These are the 2 plants which are controlled. These are the 3 plants which are inoculated. So you select for plants which have got at least a 50% increase in biomass, root biomass, more nodulation, and that leads to more nitrogen fixation. A group of microorganisms which are called as actinomycetes, which you've heard about. Actinomycetes are actually, because things are taught in a certain way, in our textbooks you'll find bacteria, fungi, actinomycetes. That's how that's how it has been taught in this book. But actually, actinomycetes are also bacteria. So actually, you could have just said bacteria. Then, in fact, they're called as actinobacteria. So actinomycetes is actually actinobacteria. And Streptomyces is one of the most important bacteria, or important actinomycetes, as you would like to call it, which is responsible for producing streptomycin, which all of you have heard of. So many other antibiotics which come from actinomycetes. But these are also plant growth promoting rhizobacteria. So this is a slide from one of my students. We isolated from the rhizosphere of various plants and then screen them for indole acetic acid production and biocontrol, whether it's against Macrophamina fragiolina or Sclerotium rolfsi or Rhizoctonia solanae. And then that's how we screen them and then use them for biocontrol. So these are very good for that purpose. This is the same slide, so we need not go further into this slide. Then there's another group of organisms which are called Arthrobacter. Now, Arthrobacter is also a actinobacteria, and if you look at the culturable fraction of microorganisms, culturable fraction. I said that 99% are not culturable, only 1% are culturable. When you look at the culturable amounts of bacteria, almost 15% of them in most soils you'll find Arthrobacter. And Arthrobacters are, we studied the Arthrobacter diversity in vertisol. This they look like. Arthro means, it's like this joint. arthroscopy you would have heard of. So arthro is joint. So the bacteria will be like jointed like this, jointed rods. Arthrobacter means rods, arthro means joint. And if you look at their morphology under the microscope also, you can see here, you can see a V-shaped bacteria. At the top. So you can, this can also be an indicator of soil fertility or soil biological health. For example, when you look at the diversity of Arthrobacter in terms of morphological diversity only, in chickpea and maize, in chickpea we could find more than 50 different types of morphotypes. In maize we could find about 30, soybean about 20, wheat, rice. So that means legumes are very important in improving soil health. We always say that, right? So here this picture itself tells you, and a C4 plant like maize has got more morphotypes than a C12 plant, C3 plant like wheat. So because of more carbon coming out, the diversity of the carbon compounds which are coming out of the plant roots. So this is another group of microorganisms we have worked with. Rhizobium we have worked with, or PGPR like Azospirillum and Bacillus, Acidification we have worked with. We have worked with Streptomyces, Actinobacteria, which I told you, and also with Arthrobacter. Another group of organisms which you must have heard of is Pseudomonas, which is a very important organism. And this is used in biocontrol. And this is a slide on groundnut. This is a slide on groundnut. There's an organism called a Sclerotium rolfsi. This causes what is stem rot. So when the stem rot happens, the groundnut plants will just fall here, as you can see. And this is a fluorescent Pseudomonas, which fluoresces. When you use that as an inoculant, so it's lush green. And this is the one which is controlled, where it has not been inoculated. And the yield of this is almost 15 to 20% more as compared to here. The same thing by control, we have also worked in hills in Solon. This is in one of our centers at the Y.S. Parmar University of Horticulture and Forestry. There's an organism which is called as Dermatophyra necatrix. This causes white root rot. So this is the root rot. So this is an apple seedling which has been affected by white root rot. You can see the white fungus. And the one which has been treated with Bacillus licheniformis. The Bacillus licheniformis suspension, you can just dip the stalks in that and then grow them in the outfield. And this is important. And the best part is that even if there is no disease, there's a program which is called as Rejuvenation of Diseased Orchards. What happens? The orchards, when they have run for a number of times, 40 years, 50 years, the productivity falls because of poor soil health, because of buildup of pathogens. There are deleterious microorganisms. There are beneficial microorganisms, there are deleterious. So to change the microbiota and change the soil ecology, you need to, either give the soil rest, or then you can also inoculate with some of these microorganisms. So our scientists have found that it is not only improving the yields of plants which are healthy, diseased orchards, but also in healthy orchards also. So you can use these inoculants nevertheless. Mycorrhizal fungi are very important. Mycorrhiza, almost everybody you must have heard of that. Mycorrhiza, well, this is a plant. I have taken the textbook from Smith and Reed. So you can see the primary root, secondary root, tertiary roots, and then quaternary roots. They form a network. So this one plant. So they connect the root networks of the whole of various plants, and they help the soil, the roots to hold the soil. You see here, the mycorrhizal plant. When you pick up, the soil is still sticking. The rhizosphere soil is still sticking to that. But if it's a non-mycorrhizal plant, when you wash it, the soil is just coming. This is a soil with this slide which I used. So these plants supply the fungi with sugars. The plant supplies the fungi with sugars, and the fungal hyphae grow 5 to 10 centimeters beyond the plant roots, and they extend to soil pores too large for root hairs. So there are certain root pores which the root hairs cannot enter, but the mycorrhiza can enter. So therefore, they are able to tap the nutrient sources which are there. Increase the plant nutrient supply, especially phosphorus, and most important is to increase the plant water supply. And that is why mycorrhizal plants are able to withstand stress more than non-mycorrhizal plants. So when the soil is healthy, it has more soil organic matter, it has more soil biodiversity. Automatically, it means more mycorrhizal activity also, and they will help. Extremely important group of microorganisms, which are again It can be used as inoculant. This slide I included because there was a lot of interest in biodynamic agriculture. There's a lot of interest in zero budget natural farming, where they use all these cow dung bio-based formations, cow dung based biodynamic. These preparations. Is there anything useful here? So what we found was high numbers of bacteria, about 10 to the power of 6 per gram, about 1 million cells. Yeasts, which are again fungi, 10 to the power of 4 per gram. Actinomycetes, 10 to the power of 3 per gram, as also high proportion of lactobacillus species. We identified several bacillus species with PGPR activity like Bacillus cephrensis, Bacillus cereus, Bacillus subtilis, Bacillus licheniformis, Bacillus dendriticus. So yes, there are a lot of beneficial microorganisms there in these cow dung-based biodynamic preparations. These are Panchagavya, also BD50, and then CPP, Cowpat 50, and many others. And there was also an earlier reference by Swain and Ray in 2009. They published before us. They found Bacillus corynebacterium, Lactobacillus, Leuconostoc, Bifidobacterium, Enterococcus, Streptococcus, Aspergillus, Rhizopus, Trichoderma yeast in cow dung. So there's a lot of interest in this. So I thought this is one slide which I included just to let you know that yes, they do contain beneficial microorganisms and can be used. Now coming to soil health. Now again, this is the fourth part of my lecture. We all know that organic amendments for years together, for millions of years, for thousands of years since agriculture has started, organic amendments have always been used to improve soil health. Cover crops, that means green manuring, no tillage, legume cultivation improve soil biological activity and improve soil organic matter formation. Why I included this slide is this was at my Parbhani center. We found a citrus orchard. This is as you can see, two citrus orchards, and they're standing between two citrus orchards. And this gentleman has been green manuring with Sesbania aculeata, or what is Sesbania cannabina actually, dhencha. It's called as dhencha. Probably it goes by the same name here too. For 30 years. So every year, and this is the longest I have seen anybody doing green manuring for such a long time. So you can see the earthworm here. Here you can see, and you can see this picture which I have taken with my camera. And Damodar, you have worked with what is also a what is all structure will never be like this. This is looking like ghee. When you make ghee, it comes out as granules, so beautiful. This is a black soil. What is all? So that means soil structure is not something which is permanent. You can transform soil structure any way you want it. It's just that it's got more organic matter. It's more porous because you're using green manuring. And it's a beautiful soil structure. The water will go inside, the soil will breathe the best activity, and you get very high quality citrus which you can exports to. So this is commercial agriculture, what you guys are going to do. So probably the one lesson I can do is for best commercial agriculture, for best quantity, just do permanent green manuring network between the orchards, which is possible in tobacco. And then export, and they are very sensitive about the export of grapes from Maharashtra. This is from Maharashtra. In case the grapes have got any pesticide residues, consignment comes back. In case the orchards of citrus has got anything, comes back. So this is nothing. There's no need to do anything. Just do green manuring, permanent green manuring, and you get a fantastic quality of that. And then this slide is though not from here. This is from Northeast. This is just showing you springtails and columbola. This is how they look like. And one could look at the faunal activity, because people don't look at faunal activity in agriculture university. Your universities which do that. Very little work on soil fauna has been done. Somebody could do work on this. Biomanures, again, these are all very well known. Enriched manures, whether you enrich with rock phosphate at the rate of 1% P and then add nitrogen fixing bacteria like Acidification or PSP. So you can use this and then prepare an enriched manure. And then another thing which you could do here is, because a lot of rice is grown here. Yeah, it's okay. Because a lot of rice is grown here. That is Azolla. Azolla, all of you know Azolla. It's a macrophyte. If Azolla is available, it's fine. If Azolla is not available, any green macrophyte, sometimes you do lemna, sometimes you do so many other weeds that you can pick up. This is from my Jorhat Center in Assam Agriculture University. What they do is they put one layer of rice, then one layer of this green Azolla, then one layer of rice, then another layer of green, and they build it up. So this is the whole pile that you see. and then allow it to make a compost. And at the end of 3 months, this is kind of compost is there. And then you can use this. While doing this enrichment, they do add this rock phosphate and NPK. So this is another intervention. So it's not just inoculants only. And if you can put inoculants in this, then all the more better for making suppressive composts, which I mentioned. Suppressive composts are those which suppress plant disease. Now coming to the indicators of soil health, I've just finished that part. While these are the most important indicators of soil health: bulk density, aggregation, and water holding capacity. I mean, if the bulk density is good, and if aggregation is soil is very good, if water holding capacity is very good, automatically you can all of these is good. If the soil physical indicators are good, automatically you can, because if soil pH is 10, aggregation won't be good. I mean, these are all related. We have broken up these into physical, chemical, and biological. In the soil, there's nothing like physical, chemical, biological. It's only one indicator. I hope. So soil is, I have written here, soil is an integrated system. It integrates physical, chemical, and biological. It is not a collection of separate parameters or processes. For example, physical and chemical properties are shaped by biological activity. I've just said that soil organic matter is formed by microbial activity. So if soil organic matter is high, aggregation will be better. So these are all related things. And biological activity is enhanced or limited by chemical and physical condition. If soil acidity is very high or soil alkalinity is very high, biological activity is very high. So although we, for the purpose of our studies, because somebody is a soil physicist, somebody is a soil chemist, somebody is a soil biologist, you break down these and then measure separately. But in the soil, there's nothing like soil physics and chemistry and soil biology. These are all integrated. So you have to measure all of them together. And if you measure soil organic matter, that's why I put it in red. This itself integrates quite a lot of activities. Although these are the most important parameters which indicate for productivity purpose. There's an element which is called labile soil organic matter. One is which is the portion which is easily oxidizable. Your available nitrogen is actually sort of an available labile organic matter for magnet oxidation. And then soil respiration. How much of carbon dioxide, how much soil is respiring carbon dioxide per unit amount of carbon. Even nitrogen mineralization. If nitrogen mineralization is good, soil quality is good. Soil enzymes are good indicators of soil. If there's a lot of earthworm activity, it's good. If the soil can suppress diseases, soil quality is good. So very difficult to actually pinpoint and say what is a good indicator of soil quality, what is that single indicator. I'll give you an example. There are strong functional bridges between the physics and biology of the soils. I've just mentioned to you that if soil organic matter is good, that means microbial activity is good. You can't have a soil very high in soil organic matter and say this biological activity is poor, because soil organic matter is building up because of good soil biological activity. If this is good, I think physics aggregation will be. So there are strong functional bridges between the physics and the biology of soils. Now, let me give an example. This is from England, the Institute of Grassland and Environmental Research in Aberystwyth, where I had visited when I was there. This is a soil which is 30 times more compacted than this soil. This is a normal soil. And you see the clover which is growing so beautifully. And you compact that by, pounding it. This is 30 times more dense than this soil. And look at the poor growth of this automatically. Right? So soil under clover, the soil particles aggregate into distinct crumbs, bulk density decreased, and water percolation improved, and there's better aeration under clover. This one. You see this here? This is more aggregated as compared to that under grassland. This is the grass, this is the clover. The soils under clovers aggregated into distinct crumbs, bulk density decreased, and water percolation improved, and better. So we'll not talk about the rest. Then this is again work using several other indicators. Certain genomic indicators of soil health, soil genomics that we worked on that. So this is from an aridisol. This is in Rajasthan in Hanumangarh. This is a farmer where I had taken soil samples, both in coffee cropping as well as in the citrus orchard. So this is 16S rDNA, which is prokaryotic. Example 2408 is using an RT-PCR. Now look at the number of gene copies. Under organic cropping, it's almost double than that in conventional cropping. And in organic orchard, it is almost again double than that in conventional. There are certain genes like NIFH, MOA, various other genes which you can amplify using specific primers. Now, these primers, you see MOA is less in organic cropping and less in organic orchards. But here less is more. Here more is more, but here less is more. Because it doesn't have to deal with high concentrations of ammonia. When urea is hydrolyzing, you get ammonia and then is hydrolyzed by urea. Urea is hydrolyzed and then you get ammonia, which is then nitrified. Here, because it happens very slowly, the amount of nitrogen mineralization is slow. Therefore, the number of gene copies are also less. So this is a positive indicator of soil health, this is a negative indicator, and both show that the soil health in organic cropping is better than that in conventional cropping. And again, I just mentioned to you that actinomycetes is also a good indicator of soil health under conventional cropping and organic cropping, conventional orchard. This is cowpea, this is citrus. Again, the population of actinomycetes is more. Glomalin, as the word glomalin is derived from the word glomus. You know, glomus word is glomus mossi is a basically avascular mycorrhiza. You know that. So this is a kind of a protein which is produced glomalin. Again, glomalin protein is more in organic cropping as compared to orchard. So these are glomalin is very easy to determine in your laboratories. Any soil laboratory you can determine glomalin very easily, and that itself could be used as a good indicator of soil health. You don't have to use genomic indicators everywhere because it's expensive to use. But certain indicators like labile carbon is a good indicator, soil respiration is a good indicator, glomalin is a good indicator. This is a slide on high input farming. This I work in certain areas of Guntur where chilies are grown and black gram are grown, and using very, very high amounts of chemical fertilizer and pesticides. Now, this is chili normal and this is chili high, where almost 2.5 times the amount of chemical fertilizer and pesticides are used. Organic carbon is almost the same in both of them. But look at the microbial carbon. That is microbial biomass carbon is less here under the high. This is about 10% less here. That means the amount of microbial activity here is less. And importantly, metabolic quotient is high. Metabolic quotient is the amount of carbon dioxide per unit amount of labile carbon. So that is the efficiency of the microbes which are working in the soil. Here the efficiency is they are respiring more carbon. Although carbon A, microbial carbon is less. Try to understand this very simply. Microbial carbon is less here. But to produce a unit amount of carbon, they are having to respire more. Is this clear? More carbon dioxide is being produced per unit amount of carbon that is transformed, and even they are not able to build up soil organic carbon. So this is showing that the soil is under stress. So these are very, very sensitive indicators of soil health, which a conventional chemical analysis would not show. Whereas here, you see when black gram is shown, and black gram is a legume, it fixes nitrogen, improves the resistance of microbial activity. We have shown there that the Arthrobacter diversity also had shown under chickpea, it was more. There, although the high inputs that were used were marginally high, because you don't use such high amount of fertilizers and pesticides in black gram as you use in chili. So when you use a high amount of fertilizers, right, organic carbon has still increased, microbial carbon had also increased, and metabolic quotient has decreased. That means they are doing less work. The microbes are having to do less work. They are having to respire less to build up a unit amount of carbon. That means this soil is more sensitive. The lesson from this was that legumes, which we already know, that legumes are important in building soil health, but it is indeed so. From a scientifically, you can prove it from these figures that legumes are important in maintaining and protecting soil health. It's basically it's the same work which I have just now summarized to you. It's the same data in slightly more. And the enzyme activity. Now this is in the case of organic cropping versus organic orchard. This is back to that slide from Rajasthan. Now this is organic cropping, this is organic orchard. As compared to the conventional one, these enzymes, which are acid phosphatase, fluorescent diastase hydrolysis, dehydrogenase activity, and beta glucosidase, are much higher. And again, beta glucosidase is a very important indicator of soil health. So if you are looking for easy measurements of soil health as the parameters, I would recommend, besides organic carbon, labile carbon, soil respiration, glomalin content, and beta glucosidase. You look for one enzyme, so you don't have to look at all these. It's for a soil biology. But for anyone who's devising a soil health program, where you want only one indicator, then go for beta-glucoside, go for glomalin, go for levothal, and soil respiration. You can do along with organic carbon. This gives you a complete suite of soil biological activities that you will require for your research work, which you can publish also. We also did what is called as soil metagenomics, because everyone doesn't have to do this. We do it because we are specialized. So when we found that legumes are improving soil health, so we said, let us look at the various phyla, bacterial phyla, which are there in the soil. So we had the same black gram 1 and black gram 2, which I've just showed you in Guntur, the one which is under normal inputs and the high inputs of black gram. This is chili, normal inputs, and very, very high inputs. So soil metagenomics, that means you take, metagenomics means you don't look at a particular organism, you take the entire soil DNA, amplify it, and then do next generation sequencing, and then look at the phyla. So these are the phyla. This is Actinobacteria, Gametomonotids, Acidobacteria, Firmicutes, Microbial, various kinds of. There are about 30 to 50 bacterial phyla in the soil. Generally, you'll look at about 30 phyla. In this, I've shown you about 15 or so. This is a scale. Each dot represents about 16 gene copies, 32 gene copies, 64 gene copies. Now, this picture, I think even a small child will tell you, there's no need to even look at these names. The very size of these dots of Actinobacteria tells you that the best soil health is here and the worst is here. So that means Actinobacteria are a good indicator of soil health, microbial health. If you can do the genomics of this, so it's the best here and it's drastically reduced here. So the diversity, and this is number one. Number two, this picture again, as I said, a child can tell you that in terms of phyla, in the phylogenetic distribution, you can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 here. And then around the same picture, and only 3 phyla here. So what does it show? Directly it shows that when you're having chili cultivation with very high amount of inputs, the soil health has gone down and the bacterial phyla has gone down. Only just 3. And not only that, the picture has just reversed. Alpha proteobacteria have increased here. And alpha proteobacteria, when it has increased, means it is a disturbed ecosystem. Whenever an ecosystem is disturbed, the alpha proteobacteria will be more, because it's having to cope with a lot of xenobiotics which it has to decompose. And actinobacteria, which are involved in building up the soil, that is there. So we'll not go into all this. NF genes they reduce, other geodramatic. We don't have to go into all this. Just this picture tells you the soil health of a situation where you have very high inputs. And then I'll take you back to Rajasthan, because we did metagenomics of vertisols in Guntur and in Rajasthan. So I'm just going back and forth between the two slides. Perhaps this is the last slide. This is showing organic cropping is the one which is in blue, conventional cropping which is in this slight brownish color here. You see these blue skyscrapers here. Actinobacteria were in higher proportion by 10% in organic farm. So here you see this is Actinobacteria. This is the second one. So this is higher than this. And alpha proteobacteria is just the reverse. So there's always inverse relationship between Actinobacteria and alpha proteobacteria. So this is again my student Apana did PhD with me, and we found that Actinobacteria were higher in proportion by 10% in organic farming, and Proteobacteria were higher by 20% in conventional farming. And also we did the plate counts also. This is genomics. And it's not enough to do just genomics. You have to have a indirect confirmation also. Actinomycetes plates counted 90% higher. So this tallies with this, that the counts of actinomycetes are also high and the genomes are also high. So then we proposed that organic amendments as ecosystem engineers. This is the first time in the global literature we produced that organic amendments. We all the time talking about nutrition point of view. We proposed that organic amendments are ecosystem engineers, microbial, biochemical, and genomic evidence of soil health improvement in tropical areas on field site. So we then concluded that microbial amendments and legume cultivation improve soil biological activity and soil organic matter formation. Just kindly note the word I have written, soil organic matter formation. I have not used the word soil organic matter synthesis. That is again a big topic, another seminar, because there is no synthesis of soil organic matter. It's only formation in the soil. Actinobacteria are good indicators of soil health. Population goes down under intensive cultivation with high fertilizers and pesticides. Small amounts of organic amendments act as ecosystem engineers to improve the Actinobacter population in biodynamically formed soils, like the one in Rajasthan that I mentioned. So this is about the Guntur paper on microbial and genetic ecology of tropical vertisols and intensive chemical farming. This was the paper on the Guntur one. So based on these two papers, we proposed Actinobacter bacteria. I now come to the word soil organic matter. What is soil organic matter? Why I said there's no synthesis in the soil? Why there's only formation? Microorganisms are the principal agents for the formation of the mixture of organic molecules that is now called soil organic matter. I've used the word now. I've put it in capital letters. So what is soil organic matter? Soil organic matter earlier uses a humic acid, fulvic acid, aminobutyric acid, and so many other things. These are all compounds which, when you extract with the alkaline sodium hydroxide and start analyzing, you get them. In the soil, you don't have, there's no direct evidence of their formation like that. In the soil, it's just a mixture of biomolecules, which I will now mention in the next slide, what those biomolecules are. Soil organic matter is the end product of the microbial catabolism and anabolism. Catabolism means breakdown, anabolism means building up, which results in chemically diverse and stable soil organic matter, whose accumulation is driven by distinct microbial communities. When you say there's a diversity of soil organic matter in the soil, it's because of the diversity of microorganisms. But sensu stricto, that means strictly, they, that is the microbes, are not making soil organic matter. They, that is the active microbial biomass, are rather their remains, microbial residues and necromass, are soil organic matter. Now remember that first slide in the textbook when I showed you that organic matter is 5%, and then it said 80% are macromolecules. And I said active is only 5%, and roots are 5%, 80% is active biomolecules. I had gone back in the first slide. So that is the 80% that I am now referring to, that the microbial biomass, which may be only 5%, but 95% is the dead remains, necromass. Necro means dead body, right? Necromass. You will never heard this word. So the necromass constitutes the soil organic matter, which then turns over slowly. Is stabilized on, of course, on clays by binding to the clay. So what are the components of soil organic matter, which you can now analyze as a very good indicator of soil health? If you've got a pyrolysis GCMS, you've got GCMS in your institute, which I saw in the morning. There's a pyrolysis unit which is attached to that. So now, I just now said that soil organic matter is not humic acid, fulvic acid. That is what you get when you extract with the 0.5 normal sodium hydroxide, you analyze it. That is what you get humic acid, fulvic acid. But if you don't extract, you just make a water extract from the soil or with a buffer. What is soil organic matter? Basically, about quarter of that is protein, 24% or 25%. This is a slide from Kallenbach 2016 in Nature. About a quarter of that is protein. Polysaccharides are about another 27%, or a quarter. So 50% of soil organic matter is proteins and polysaccharides, sugars, hexoses, various things. Lipids are about 5%. Phenols and aromatics, which is about 10%, which is the constitutions of humus when you go for various things. There are nitrogen-bearing compounds which are about 14%, and there are unspecified compounds which are about 16%. So these are the 6 fractions into which soil organic matter now can be analyzed using a gas chromatograph. So in the terms, this was my emeritus project, my final project which I did on microbial strategies to improve soil health. When I had worked for the last 15 years at Bhopal and then indicated that actinobacteria are important in maintaining soil health, we have genomic evidence. Then can we use them as inoculants now to improve soil health? So these are Arthrobacter, these are Streptomyces, which I have just mentioned. I use them as inoculants, and when you inoculate, this is Hilton-Milner broth, and then grow them for several months in these flasks, they produce the humus-like compounds. The brown humus-like compounds that you see in the soil, these compounds are also produced in the media system, uninoculated medium, and then when you inoculate it, and then I have E4, this is E3 by E6 ratio, E4 by E6 ratio. These are various ratios which are used to characterize soil. So we had this uninoculated Arthrobacter species, Streptomyces species, Bacillus species, and then you mix all of them together, and you see there's a reduction in here. Whenever there's a reduction, it means this formation of more heterocyclic compounds, like humus-like molecules. So melanoid pigments produced by metabolism of phenolic and indolic compounds, high amounts were wherever Arthrobacter is included. Then we studied the enzyme activities which are responsible for the transformation of these compounds. So interventions like inoculation of microbial consortia improves the microbial activity, microbial biomass formation, translates into higher amounts of labile carbon, soil organism and better soil health. We then use these inoculants in farmers' fields for growing maize, wheat, chickpea, and consistently we got 10 to 15% increase in growth yield. I'm not talking about those results. So whenever we we incubated with wheat straw and soybean straw, and then we measure various parameters as I mentioned. Carbon mineralization means respiration, pH, organic carbon, labile carbon, and then labile carbon. This is carbohydrates, extracellular protein, dehydrated activity. And this last one, where you use wheat straw and soybean straw and inoculum, this has given the best result. Now I come to the final few slides of my presentation. Sometimes you don't realize the importance of microorganisms, which I've just mentioned, that they are not only important for plant and soil health, but also important for animal and human health. 78% of the antibacterials approved during 1983 to 1994, they have come from microorganisms. All the antibiotics that you have used. Beyond antibiotics, you go 40% of all prescription drugs, whatever prescription drugs you get in the hospitals, have their origin in soils, including 60% of all newly approved between 1989 and 1995. 60% of new cancer drugs approved during 1994. These are all from the soil. And therefore, this is the importance of maintaining soil in a pristine form. If you degrade your soils, we are also degrading human health, because the source, as you know, you must have all heard about this, that we have virtually exhausted all the antibiotics that you can get from the soils. Over the last 40, 50 years, there are no more new antibiotics coming from soil. Some of them are synthetic, but new antibiotics have not been discovered. I don't have this slide here. I think the last antibiotic was discovered 25 years ago from soil. And the latest one, I forget the name, I think Ticobactin or something like that. That was the last one, which is by a new isolation technique that was discovered about 5 years ago in Boston. So investigations on the ecology and life cycles of human pathogenic soil organisms and the influence of climate change on soils and human health is very important to study this. So the sustainability paradigm, when we talk about soil stewardship, starts here with a healthy soil, which gives you a healthy plant, which gives you healthy animals, and this leads to a healthy planet. So our ecosystem starts and ends with soil. And that's where that's most important. That's where ecological stewardship is coming from. And if you look at even Atharveda, the very quotation around 3,500 years ago, 4,000 years ago, what of thee, O earth, I dig out. That means whatever I'm taking out, what of thee, O earth, I dig out, let that quickly grow over. Let me not hit thy vitals, nor thy heart, O cleansing one. That means in this quotation itself, it has been taught to us that soil is the one which is cleaning, O cleansing one. And let me not hit thy vitals, nor thy heart. That means they realize that there is something in this soil which is vital. That is, could be microbes, it could be organic matter, that whatever you want to say, vitals, earthworms, whatever things, that I should not hurt the soil. This was known that. And whatever I take out, let that quickly grow over. That means sustainability is there. That means whatever I take out, let that should be able to quickly grow, and we should not do that. So all of you, I don't know how many of you heard, there was a summit called as Vaibhav. This is Vaishvik Bharatiya Vaigyanik Summit. You know, this was a global summit of overseas and resident Indian scientists and academicians in October, November in 2020, which was taken with the initiative of Prime Minister Modi. The leading intellectuals from all over the world interacted with Indian intellectuals, and to identify technology and knowledge gaps, to identify futuristic areas of research, give impetus to objectives of Atal Innovation Mission, strengthen Atmanirbhar Bharat Initiative for high-end research, bring out the mechanisms of advancements in education, research, and entrepreneurship in India as an essential element of its overall sustainable development. So there were 16 horizontals in various areas. In agriculture, there was only one horizontal, that was horizontal 2 on sustainable and climate smart agriculture. There was another sub-vertical in that microbial resource sustainable agriculture. This was shepherded from ICAR-NIRCA, but steered from ICAR-NIRCA. And then there were about 4 speakers from abroad and 2 speakers from India. So I led the lead lecture from India on building partnerships for soil ecological stewardship, biological nitrogen fixation, microbial inoculants, and soil biological indicators. Much of what I've spoken today is from that lecture which I had given in the Prime Minister's Summit. This is what I had spoken, and what I had pleaded there was this, that whatever we want to do and whatever I have to do, I cannot do everything myself. Whatever I knew, I have done with the help of my colleagues all over India in the various coordinated research projects over a period of about 25 years. So for that, we need centers of excellence. So we proposed Indian Nitrogen Fixation Research Center, like I headed the UNFC for the last 5 years before I retired. Indian Microbial Inoculants Technology Center, Indian Soil Ecology Research Center, IRA. So that these kind of institutions should be built up in IRA, NBI, MAO, and ISS Bhopal, and with collaborations among all ICR institutes and commodities institutes and state agriculture universities, and strengthening the frameworks of the already existing networks of the crop and commodity ICRs. Only this, we require good funding and research. We have the capacity to deliver. We have the scientific strengths and caliber to deliver. We need institutions, networks, and more money to do this. So, in conclusion, food production, mitigating climate change, and ecosystem services. That is what we talked about today. They're all closely intertwined and all rely on soil biodiversity. We need to shape agricultural practices that builds on soil biodiversity in order to maintain or improve the soil ecosystems of the future. Maintaining the soil ecosystems of the future. There's a Future of Soil Science, a publication brought out by International Union of Soil Science in 2006. This is the sculpture of Rodin, the thinker, and I had contributed to this. And this is the book, The Soil Will Save Us. This is a book called by Christine Olsen. And one of the chapters by Ratan Lal, and which he had presented to me when I went to visit him. That ultimately they have all come to realize that the soil is going to save us if we maintain everything in a pristine shape. So thank you very much. It's been a great pleasure to speak to all of you. And I hope that if a little part of my message has gone across and builds into research programs here, that will be a great pleasure and payback for my coming here. Thank you, Damodar. Thank you very much. If you have any questions, I'll be very happy to answer. Yeah.
Thank you very much, sir, for your great lecture. It is very informative and thought-provoking. I request all the participants to interact with sir.
Commercial sources, you don't want to come. Okay, fine. Otherwise, there are good commercial sources. It's not that they are bad. There are good commercial sources which supply. Even on the net also you can find them. On Amazon, you can find. Just try out. It works. Otherwise, I could have suggested Terry, New Delhi, Terry, Tata Energy Research Institute. I will email Dr. Reddy. Whatever email you send, me just copy. Thank you. Because I'm not sure about Amaravati whether they have got or not. Tamil Nadu Agriculture University should have. Yeah, I don't think that